what these types of changes that we're seeing either in DNA copy number sequence, looking at chromosome modifications or gene expression, what this actually means in the context of looking at normal function and then obviously in the function of genes in a disease setting. So the model systems that you can conduct functional genetics are obviously as broad as all of the different model organisms that we have available to us. On a gene-specific level, you can do a lot, or even on a large scale, you can do a lot of these experimentation in bacteria, yeast, C. elegans, zebrafish, Drosophila, and mouse. In the context of human, most of the work I'm going to talk about today has been done in transport cancer cell lines. And I'm going to give you a hint of some of the advantages and disadvantages of that as we go through. Increasingly, though, particularly with some of the new technologies, you're seeing it being able to be applied to primary cells, into patient-derived xenografts, into PDXs, and then induced pluripotent stem cells. As I say, what I'm going to talk about predominantly is in cell lines, but it can be extrapolated across. Okay. So there are really sort of two flavors of functional genetic analysis that I'm going to talk about. I'm going to predominantly talk about the second of these, but I just wanted to mention this first, which is the gain of function. So it's when you're overexpressing or you're sort of replacing a gene to see what its effect is in a different context. So you can overexpress a wild-type gene. Make sure I get it. You can overexpress a wild-type protein in a mutant background to see what the functional effects are of that. You can use overexpression in epistasis tests where you can overexpress different genes and look at how that affects a particular phenotype. Where there's actually probably been most work is in the overexpression of a gene that usually there's open reading frame of cDNA in a heterologous system, either in a whole organism or in the cell lines that I described earlier. And these can be used to look at wild-type proteins or mutant proteins. They can be used to create interaction networks, and they can be used as modifiers for screens or looking at drug targets. So there's really a very large way and different ways you can use these types of overexpression studies, but there has been a tendency to actually not do this on a large scale. This is hard to manipulate, and it's been hard to read the phenotypes from these. So there's been most emphasis in the functional genomics field really on a loss of function, where there tends to be a clearer phenotype at the end of it. So there are two types of loss of function in the crudest type way that I'm going to look at. You can have a knockdown, or you can have a knockout. Okay? So I'm going to talk about both of these, but they are different. Okay? So when you knock down, you've often still got a residual level of protein expression, or certainly an mRNA expression there, and that you really need to think about that in the context of the phenotypes that you're investigating. A knockout, in theory, should take out all of the proteins or all of the RNA molecules that you're targeting. So you have a difference that needs to be brought in there. The other piece of this puzzle that I just want to put forward is the difference between forward genetics and reverse genetics. And then I'm going to pull all of these together to discuss the different technologies that are available to us. So forward genetics is the mapping of a phenotype to mutation with no prior genetic information. And for a long time, this was done in the context of mutagenesis studies using chemical, radiation, or viral mutagens. The other is to do reverse genetics, where you're using prior genetic information to generate the reagents that induce a phenotype. So for a long time, and particularly in mammalian systems, we needed to do forward genetics because we didn't have all of the information we needed to do reverse genetics, which for a long time was really the purview of things like yeast or Drosophila models. But obviously with the identification, the full sequencing of the human genome, and then with the tools that I'm going to talk about today that we've developed, increasingly we're now able to do reverse genetic studies. So I'm now going to sort of move on to the technologies in the context now just of loss of function, okay? So they really come in two types. Those where you're focusing on gene silencing, which is often, as I say, is referred to in the literature as sort of a knockdown, and is predominantly using the RNA interference mechanism. The alternative, which we've now seen increasingly now become applicable, is to do gene editing, where actually this can be both either a knockdown or a knockout, depending on the effector molecules that you're using. And these come in two predominant flavors in terms of technologies that have now developed over the last few years. One's using transcription activator like effector nucleases or TALENs, and then the ones that obviously use clustered regular interspaced short palindromic repeats or CRISPR. And I'm going to go through these in a little bit of detail just to give you some basics. Now, there has been a plethora of different reviews over the last few years that have been looking at these different technologies. I'm going to focus on one that came out actually now a couple of years ago, but really is still relatively, how can I say this? It's very relevant still. A lot of the issues haven't changed that I really want you to focus on. And the key thing here is, and why they've titled it, was choosing the right tool for the job. And it's the concept is that actually what is really great now is we have options. We can try all of these different things and we can see which is going to work in the context of a particular experiment. So I'm going to walk through these three different main areas. I'm going to talk about RNAi very briefly, then go on to TALEN, then go on to CRISPR-based techniques. So RNAi is a post-transcriptional mechanism that induces RNA degradation. And so this is a reversible knockdown. And the major ways of inducing the effect is to use a small interfering RNA. So the RNA mechanism is a naturally occurring gene silencing mechanism. It's predominantly known in the context of using microRNAs that regulate post-transcriptional gene silencing by interfering or interacting with the 3' sequences of a transcript, and that alters the expression of a protein from a particular transcript. But we've been able to exploit this by using siRNAs or shRNAs that actually target specific transcripts and induce a degradation of that transcript. Now, one of the issues has been that there is quite a large off-target space for this. Effectively, because of the way that microRNAs work, you can have an interaction with an siRNA with a lot of different sequences, so that has been one of the major problems with this. I'll mention how this can be overcome in a few minutes. But it's still very robust, and it's very quick in comparison with CRISPR. You could do an siRNA experiment in a couple of days. We're still doing CRISPR experiments that's going to take you a long time. So sometimes a lot of the question is simply, what is the question I want to know, and how quickly do I want to know it, as to why it would make sense to use an RNAi-based experience. So just to cover what I just mentioned in terms of how this works, so predominantly, we're using siRNAs or shRNAs. So siRNAs are synthetic molecules that can now be purchased from many different commercial sources using fairly robust algorithms. Typically, one will test at least 3 to 4 different siRNAs per gene to be sure of successful knockdown. shRNAs are going to be expressed from a plasmid, and they can be expressed also from an expression vector. This is targeting, then, instead of the RNAi pathway. I'm showing a highly simplified version of this, where I'm just focusing on the cytoplasmic part of the pathway, where it's going to interact with the RNA-induced silencing complex, predominantly with the protein AGO2, which will take one strand, it will take the guide strand from the siRNA or from the shRNA that's been processed by DICER. It will align that on a sequence base, and it will cleave in the middle of this. This will reduce protein synthesis as a consequence of the fact that you have degradation of the transcript and therefore then a loss of function. So the assays that can be conducted downstream of this are fairly obvious. They're going to be mRNA, protein, or function. And what really has to be optimized to apply this technology is making sure you've chosen the correct model system to be able to look at your particular question related to loss of function, the efficacy of the particular reagent that you're using, and then, as I mentioned, specificity. Now, the TALENs are sort of an in-between stage, if you like, between RNAi and CRISPR. And what they're using is a way of inducing a frame shift DNA mutation, or as I'll talk about in a different way, you can actually repress transcription, which sort of becomes a little bit more like RNAi. When you're inducing the DNA mutation, obviously this is a permanent knockout, whereas if you're using an effector molecule that induces a repression of transcription, this can be reversible. So the required information for this is the transcriptome, which is the same as with RNAi, but what's key for tail of repression is you actually need to know exactly where the transcriptional start site is for your gene, and that's not always very clear, and it's not always very amenable. So the off-target space for this is thought to be a little, particularly for gene editing, is thought to be a little less, because as you'll see, you need a lot of sequence to define where you're going to edit, and that should reduce the off-target space. But you can also, in the context of repression, you can see that that may not be as specific as you would like. I should have stressed this here in a minute ago. So many genes have transcript variants, and that's one of the things you really have to consider when you're designing these experiments, that if you're going to be editing a gene or silencing a gene, are you going to want to take out all of the variants, one subset of variants, because these variants may have different functions. And so that might be come into your thinking. All right, so I'm going to talk a little bit more about tails or transcriptional activator-like effectors that can be used for programmable DNA binding proteins. So these are TALENs, and they use synthetic proteins that are based on the protein script secreted by the Xanthomyrmenas, which is a gram-negative bacteria that can infect a wide variety of plant species. Okay, and so these have DNA binding domains, and that's what's absolutely key about these. So they contain 7 to 34 homologous direct repeats of these 33 to 35 amino acids, and these can recognize a specific DNA motif that is also still present in a mammalian. So the specificity of each of these tails, each of these repeat sequences contained within the 2 amino acid positions that are 12 and 13 of each repeat. I'm going to show you a picture in a minute, so we'll make more of this.